Very much, Florian Fischmeister, for this kind introduction. My part or my responsibility will be what comes afterwards, after the lab work, when you have data. And I will try to focus on the challenges of these data types. So, just to give you a brief overview, what I will tackle today is first a little introduction about data types, then the different an example, because I thought it's easier to understand our hassle with data when we have some examples. So one is for compositional data, then extreme examples, so the most complex thing that you can do with microbiome analysis, multi-omics. Then something from our lab, so our main experience that we have with DNA-based analysis. And at the end also I want to have some words about our project that we have here in the frame of the BBS Spring School. Okay, let's talk about data types. So microbiome data can be quite complex. First of all, file sizes. So now we are in the days where we can basically sequence anything everywhere at any time. And so this is a common picture where we see the costs per megabase, and this really dropped a lot, especially through the Human Genome Project. And nowadays it's not a big challenge anymore to sequence anything, it's more the challenge to analyze this ton of data and to make some sense behind the data that you have. High dimensional data for sure. So here is just a picture from a publication that tried to discuss the term microbiome, because it's not that easy to understand what belongs to the microbiome. So especially medical doctors like to use the term microbiota. Microbiota are mainly just the cells. So all the cells that we have, bacterial cells, archaeal cells, protist algae, fungi, so everything that is on a microbial level. But the story doesn't end here when it comes to the term microbiome. And we also include the so-called theater of activity. Because for sure these microorganisms, they have certain structural elements of their cells. They also code on DNA level, on RNA level. This information can be also transferred, so we get to the external level now. So we have mobile genetic elements, proteins can be secreted, metabolites can be formed, toxins and so on. And this is all dependent on different environmental conditions. So the picture really gets more and more complicated when you try to understand the entire microbiome. Also it's very often we have to deal with zero-inflated data. So we have a ton of different observations. You can imagine several thousands of different microorganisms, and only a few counts or a few ones are there in a certain environment, or a few genes are expressed in a certain environment or certain condition, and we have many, many zeros in the data set. And the data set is also compositional, and this is something that I will discuss in an example later on. Little outlook into the future. The data will get even more complex. I have here some pictures, for instance, from the American Gut Project. So that's a project, it's a citizen science project, also where people without any scientific background could participate, and they really got a ton of different samples also around the world, despite the fact that it was named the American Gut Project. There are also a lot of samples from Europe, for instance. So really a ton of data. In the future, more and more of this global microbiome profiling studies will come out. So we really have a huge data set. This is just a picture where you see the Earth Microbiome Project, another huge project that tried to sample any soil that you can get your hands on. And here is the American Gut Project. So you could simply see here by this difference in this principal coordinate analysis that those samples are quite dissimilar from those from the American Gut Project, for instance. We also see this longitudinal impact more and more. So due to the fact that it's cheaper to sequence, we also can include time series of the data. And especially we see here, for instance, what happens before and after surgery or before and after you take antibiotics. And this is then also increased with metabolic data. There are also emphasis from new companies where they try to build a toilet that is like actively sampling your stool droppings, and this will also lead to a ton of data, so continuous sampling. This is also something for the future, and for sure also technologies like nanopore sequencing, they have the advantage that they are, can build their devices very small. So there's one prototype where you also have this little module, this little sequencer that you can plug in into your smartphone, and then you can sequence anything. When you're traveling around, for instance, you can sequence anything, and this will also increase the data and make everything more complex, but also more interesting. Okay, let's get to our first example, and I, like in my little introduction, I finished with the observation that the data is compositional, and I want to explain what this means in a few slides. So the main problem here is that we can't really distinguish between absolute abundance and relative abundance or proportions, so we don't know the absolute abundance. Here's one example. So when you have a population of different microorganisms and you sequence them, you get information in this direction, but it's not valid to make a direct comparison in the other direction, because that it's here explained on the other side. Let's imagine we have 3 samples, and we have 2 microorganisms in this population. So let's say the long one and the little cocci one. And you see that the total bacteria are always different, but when we sequence the data, we only have this proportional view, and then sample 1 looks almost the same, but sample 2 and 3, that is really different. When it comes to species A, for instance, would look like the same. And this is a problem, right? Here also, there's also summarized when we compare, for instance, sample 1 and sample 2 and the species A and B, that there are differences in what we see in the actual numbers that we don't know, and the proportions that we get out from the sequencing device. So that's something that we have to tackle with some fancy statistics. Or another example here, I think maybe that's, if you didn't get it now, this should help you. So we have 2 ecosystems, for instance, and in those ecosystems, only these green microorganisms are different in their absolute abundance. But when we make a relative abundance plot like this pie chart, not only this green area, so this green bug, will change their relative abundance, but all others are also dependent on that. And that would lead to a negative false assumption here. And what also is a problem when it comes to compositional data is the sampling fraction. So another example, we have 2 groups. 2 groups mean a different group of participants, for instance, and we have 1 subject, so 1 sample. This 1 sample would lead to 1 ecosystem, and we have 2 different species here, and this is what we sample. We can't usually sample each or every molecule in an environment. So what you see here is the other example with subject B. The ecosystem, like there are many, many more microorganisms, but the sampling fraction is the same, like when it comes to the depth. So this is shown here in the table. So here, the library size, so we also call this the library. The library is the thing that gets into the sequencing machine in the end. So here we have the same number, but when it comes to the microbial load, it's totally different. So that's also something that we have to consider. And as mentioned, this is a known problem, but there's also a plethora of different tools out there. So this is just a study that is profiled how often these different tools were used over the couple of years, the last couple of years. And you see, this is also, yeah, you just can imagine that we're just with this one problem, or let's make it two problems, we already have several different bioinformatic tools that can address that. So we have something like standard methods like non-parametric or parametric statistics, but we also use linear regression models, machine learning models, deep learning, and such stuff. This gray shaded area is just if the microbiome is considered as a dependent or independent variable. To increase your interest maybe even more, I want to give you some extreme examples. So the top that you can reach, let's say. So the dream or the nightmare of a data analyst in microbiome research are so-called multi-omics. So this term summarizes different types of biological molecules that you are investigating. So that could be genomics, epigenomics, so if how the DNA is methylated, transcriptomics, that means which genes are expressed, proteomics, which are translated into proteins, metabolomics, and so on and so forth. And also the combination with other techniques that are not like the classic omics things are like fluorescent activated cell sorting, for instance, or radiomics, brain scans, clinical data. So it's really a complex data set. Then you have different populations. You try to cluster them. You want to do predictions. You want to look on certain biomarkers. So you really get a multi-layered network in the end in different conditions. And yeah, so it's really not an easy task to do such an analysis. There are typical challenges. So it starts already with the data collection. So usually the data is not uniform. We have missing values. You also have to imagine that the depth of analysis is totally different. So we can go really deep when it gets to genomics, but in comparison to proteomics, we can just maybe target certain proteins due to the different chemical properties. So it's not that easy to really have this very broad view. We already need to know somehow what we want to look for. A lot of computational challenges and also lots of noise that we have in the data set. And that's not easy to really identify the signal then. Something about the statistical methods for sure, so how to bring all this data together, and also the problem with some meta-analysis, so bringing those into context with other data that you just recorded through a questionnaire, for instance, or through other measurements in the clinics, for instance, and the community distribution. So it's also not that easy when, like, there's for each of these omics, we usually have a standard, what we should record from them when we want to design such a study. But for the multi-omics, there's not really a standard out there yet. And it's also not that easy to make like a data mining on a multi-omic level, because we have very specific databases for each of these omics. And therefore, it's hard to bring them into a joint data set in the end. Some more pictures. So just this is an example from such a multi-omic study, and you see that they are not showing always the same picture. That's the big problem. You always get some different perspective from the underlying truth, I would say. So when it comes to the signal or the error that you get, how many features you get out. So difference like proteomics or metabolomics are often at the lower end. And like the different genes, the expression level is also different if you use different methods, and the statistical power when you look for biomarkers or so. And yeah, when you look here, for instance, you get this different area plots. So when it comes to taxonomy, RNA, proteins, metabolites. This is a study about IBD patients and the comparison with ulcerative colitis and Crohn's disease, for instance. So these pictures or figures should just illustrate how complex the journey to the final publication can get if you record more and more data. And in the end, very often we end up with such networks that are, let's say, interesting to look at, but maybe not really helpful to understand at the first glance. So you put all these different types of analysis in it and color them differently through correlations, and then try to make some sense out of it in the end. Okay, so now I hope you're interested in microbiome data, because it's cool stuff and it's quite challenging. But what I want to give you as another example is just our experience. So what we know, what we can do, and what is our daily life. And these are mainly analyses that are based on one biological molecule, the DNA molecule. There are two different ways how we can analyze this molecule. One is the targeted approach. So Christina Kumpic already introduced you to the 16S RNA gene, maybe. So we can do so-called amplicon analysis. And these are just the different programs that we use for that, so maybe not that interesting for you. Or we can do it in an untargeted approach that would be shotgun metagenomics or real-time sequencing technologies like nanopore, for instance. And what are, let's say, the new hot stuff on the research market are so-called prediction tools, so that you just measure, for instance, DNA, but through the help of a database, you can then infer maybe which metabolites are produced by these microorganisms. Yeah, so in each part of this data analysis, when it comes to DNA, we get some kind of sequence data. So a typical file format are FASTQ files. That means we have a DNA sequence and the quality information from the sequencer. And then we want to transform this into a so-called observation per sample matrix, or a biome table, biological observation matrix. So these observations, in our case, are in most cases different names for microorganisms, or different genomes, or different functions, or it can be also different metabolites, for instance. So let's have a closer look how 16S RNA gene amplicons, how this analysis would work. Here you see the representation of different microorganisms. So that's the genome of different microorganisms. So we extract the DNA, and then we look at a specific marker gene for each microorganism. So usually the 16S RNA gene has 9 variable regions, and we design primers that are binding in a conserved region and then run into this variable region. And this is like a barcode later on for us to identify from which microorganisms we got this part of the gene. So the basic workflow for this would be, we have sequencing, we need to trim and filter a little bit, so it's always good to get rid of the bad information, so that we also just base our final interpretations on good quality data. Then infer the sequence variants. That means that we try to figure out which was the original sequence after the PCR. We assign the taxonomy, we do some normalizations, and then the interpretation according to diversity or some insights into the ecology of the sample. And one tool I want to highlight here is this data2 algorithm. That's really a core element because that's responsible for identifying these original sequences in the data set, and this is done through an expectation maximization algorithm. So that means that the program learns the error profile from the sequencer, and just by doing this many times, it really comes up with the best solution in the end, and can tell you which is the original or the best sequence that you want to use for the annotation later on, for instance. Yeah, there's also another thing that might be worth to mention. They are so-called chimeras. So because due to the nature of the 16S RNA gene, because there are always these conserved regions as well that are very similar between the different microorganisms, the primer can bind, but through some problems during this polymerase chain reaction, we have an aborted extension. And then in the next cycle, a mispriming event can happen, and then you get this kind of chimeric sequences, and they are for sure nonsense information, and therefore also the program needs to get rid of such information. Yeah, this is just the different way analysis path in amplicon sequencing. Now in comparison, shotgun metagenomics, for instance, so that means here you have the same picture again with the amplicon sequencing, but here is shotgun metagenomics. So the main difference is that this is untargeted. We just sequence anything that we get our hands on, any DNA that we can find. So we get lots of different bits and pieces here from the different genomes. So over the full genome, you get a certain coverage on different regions. And therefore, this makes the entire process much more complicated to analyze, but you also get much more information out of this. And one tool, or there are two ways how to do that. Either you do it just gene-centric, so that means that you just analyze each of these little bits and pieces by itself through the help of a huge database. So a program that can run this relatively quickly is Diamond. So you have the entire NCBI-NR database. So that means everything that is sequenced on the entire planet or known on the entire planet. You run this and then you visualize this with a software called Megan6. And the other way, I would say the more sophisticated way, is a genome-centric approach, because you have to be aware of that if you sample something and you chop up the DNA and so on, then you get a homogeneous soup of DNA molecules, but in reality or in the actual ecosystem, you have genomes and cells that are around those genomes and so on. So it's not really just a mashed up soup. And genome-centric approaches try to bring this structure back into the original state, I would say. There's one pipeline I can just quickly walk you through. So again, we have some kind of quality control step. Then we have an assembly step. So that makes, out of these little chunks, make larger fragments. Then we can sort those larger fragments through a process called genomic binning into the original configuration. So it groups the different genomes, and then we hopefully can also close them and annotate them in the end. So I think I can skip these details for you here. So we lose something through the quality control, that's for sure. We often have also in our case the problem that we're working on the microbiome of humans, and therefore we also get a lot of human DNA through the sequencing, and this is something that we not always want to have, so that we have to remove afterwards. This is some illustration of this assembly process. So we have this chaos of different reads. We have to bring them into or reassemble those longer contexts, and this is done through so-called De Bruijn graphs. So you mesh up the individual sequence in little word sizes, so-called k-mers, and then you make very complicated, or the program does that for you, luckily, assembly graphs. So that means that it will just try to, with these different word lengths, to make the longest stretch before it enters the same word again. and then you come up with the best solution or the longest stretch of DNA. Yeah, then the genomic binning. So when you have these larger fragments, then you really want to group them into the original genomes. You see here just by the number of different MAGs, is the term for metagenome assembled genomes, that there's really a trend, and the programs are getting better and better, and more of these genomes can be recovered from metagenomic data. Yeah, usually we then get something like this. So we plot the completeness to the contamination. Completeness means if the genome, so each circle represents a genome here, is complete, and how or to which level or extent it is contaminated by other fragments. So we get so-called draft genomes out of this. And this is done by vertically inherited genes, so-called marker genes. Yeah, if all of them can be found, then this completeness value gets up. If there is something that shouldn't be there, it will increase here in the direction of the contamination. Yeah, and then for sure we want to know how often we find this genome in the data set. This is also not that trivial, because you also have to consider that each genome has a different length and so on. So you usually work with median coverages to do that. Then the annotation, so we need to predict genes, we need to predict pathways, and we end up usually with such an overview. So the most important pathways is if they are present or absent in a genome. Yeah, when we have the genome level, there are tons of different programs, and there are also, again, new environments. So this is just an example from Envio, for instance, which can give you this nice circular maps. So where you see the coverage on different regions in a genome, and you can compare different genomes. But yeah, maybe the best thing is here, you get an entire analysis network, again, from this level onwards. And yeah, there are also other tools. So when we have this, when we put the information back together into the configuration of the original genomes, we can for sure also work on so-called genome synteny. That means that we can look on the gene, not just the gene itself, but also the neighboring regions. We can compare if this gene is organized in an operon structure. So that means that it's translated or transcribed together with other genes. And yeah, so there's much more to do here as well. And as already mentioned, something rather new is so-called prediction tools, so metabolite prediction tools, or reverse ecology. It's called through flux balance analysis. So we feed in the information from the genome, and we have the information from metabolic models, so they are pre-calculated, and then it comes up with a model for the entire community, so thousands of different microorganisms. And then it will estimate through the help of so-called, through a medium, so it could be, for instance, nutrition information, so just what can the microbes use in this environment. And this will limit which microorganisms can grow faster or slower, and like limits the space where our microorganisms can excrete or import or export certain metabolites. Yeah, and you end up with something like this. Like these are different growth rates, and these are different fluxes. So how the metabolites, if they're either taken up or if they're exported. Yeah, that's the end of the theory, and I hope that it was interesting for you. I want to have a few words on the project that we put on the bbSpring web page. So Christina Kumpic already started to introduce this to you. And the overall question was here if the microbiome can influence our behavior or our personality. And maybe you think that's total nonsense, but there are already studies out there that could show. So here you have different microorganisms, and they can secrete metabolites that have neuroactive regulation functions. So it could be either oxytocin that increases the social behavior of mice. So most of these studies are based on mice, or gamma-aminobutyric acid that can reduce anxiety or depression, for instance. So we have these metabolites, and they can really sit, like the microbiome sits here in the gut, and really can interact through these neuroactive compounds with the vagus nerve that is then in this hypothalamus pituitary gland through the different activation of certain hormones. For instance, cortisol can be secreted, and this then acts back on the gut and on the microbiome. So this is really a feedback loop. Something is happening in our guts. It gets signaled to the brain, and then there is also a response from the host organism, and then the microbiome might be changed again. So in the context of this, the ocean rest study was designed. So these are the slides that Chrissy already showed to you about the gut-brain axis. So we want to know if there is a connection here as well, and especially when it comes to personality. We already analyzed the microbiome and data, and also included this guide questionnaire, but we did not include so much the psychological parameters for now. So highlighted here in green is the stuff that we already included in one of our publications, but what is really missing are the brain scans and personality scores and questionnaire information. So we have those brain scan information, the personality through the ocean test, multi-omics, let's say, from the gut microbiome. So through stool samples, we have amplicon data, shotgun data, and also metabolomics, and also predicted metabolites and food questionnaire. So really a complex data set that is just there to be further analyzed and brought into context. Yeah, with that, I would just want to acknowledge our group and Christina for the kind introduction. And I don't know if you want to show us pictures later on.